Hello everyone, I am very pleased to share with you our article published in iMeta, titled Peripheral Blood Immune Profiling and NXP3 as a Novel Biomarker for Risk Prediction and Targeted Therapy of Hypertensive Brain Hemorrhage. Intracerebral hemorrhage, ICH, is a severe complication of hypertension, associated with high morbidity and mortality rates. Currently, there is a lack of personalized diagnostic and therapeutic strategies, especially for early risk prediction. Therefore, there is an urgent need to develop more refined diagnostic tools and targeted intervention strategies. Immune and inflammatory responses play a crucial role in the progression of the disease, making immune modulation therapies a promising strategy for diagnosis and treatment. We analyzed immune dysregulation in ICH patients. Our study revealed that excessive inflammatory responses, neutrophil activation, and T-cell dysfunction are the major features of ICH. Additionally, we constructed an ICH risk prediction model and identified NXP3 as a potential therapeutic target. Molecular docking and molecular dynamic simulations indicated that dihydroergotamine, DHE, has a strong binding affinity to NXP3. In vivo experiments showed that DHE can reduce the incidence of spontaneous ICH and modulate immune-inflammatory responses. These results support DHE targeting NXP3 as a potential therapeutic strategy for hypertension-related ICH. First, we performed standard transcriptome sequencing on peripheral blood samples from 130 hypertensive patients in 2 cohorts. Differential expression analysis showed that upregulated genes were mainly enriched in neutrophil degranulation and inflammatory response pathways, while downregulated genes were associated with leukocyte and T-cell activation, suggesting neutrophil activation and T-cell dysfunction. Furthermore, we conducted single-cell RNA sequencing on peripheral blood samples from patients with hypertensive brain parenchymal hemorrhage, HBP, and mouse models of arterial ligation, AL, and ICH. Neutrophils were the most abundant cell type. Compared to the HBP group, there were shared transcriptional changes across different cell types in the MB group, including upregulation of G0S2 and downregulation of CXCR2 and CX3CR1. T-cells showed downregulation of NKG7, PRF1, and GZMB, which was consistent with the bulk transcriptome data. We applied a cell classification integration framework. Using this framework to analyze human single-cell data, we found that high-risk neutrophils were predominantly present in MB patients, while low-risk neutrophils were mainly found in HBP patients. The UMAP plot clearly separated these 2 cell subpopulations. This finding was further validated by mouse single-cell data and human transcriptome sequencing data. Previous studies have shown that neutrophils can differentiate into 4 distinct states under different cytokine stimulations. We found that, compared to low-risk cells, high-risk neutrophils had reduced polarization towards state A, suggesting a lack of stimulation by IFN1, IFN, and IFN in their immune microenvironment. Further analysis revealed significant upregulation of state A marker genes in low-risk neutrophils. Additionally, we reconstructed the differentiation trajectory of neutrophils from low-risk to high-risk. Early differentiation stages were enriched in innate immunity and viral defense pathways, mid-differentiation stages were enriched in RNA splicing pathways, and late-stage differentiation was characterized by neutrophil degranulation and inflammation. Scenic analysis revealed upregulated and downregulated transcription factors in high-risk neutrophils. In our study, patients were divided into training, testing, and validation sets. We used a multi-machine learning integration strategy to evaluate the predictive ability of genes associated with neutrophil differentiation for ICH risk in hypertensive patients. The optimal model had an average AUC of 0.977 in both the testing and validation sets. This model identified 7 key genes, all of which were upregulated in ICH patients. NXP3 had the highest coefficient in the model, highlighting its potential role in ICH. Our experimental results validated the significantly elevated expression of NXP3 in neutrophils from the ICH group. Molecular docking of over 2,000 FDA-approved compounds revealed stable interactions between DHE and NXP3, involving multiple forms of interaction. Molecular dynamic simulations showed that the root mean square deviation, RMSD, of DHE remained around 1.2, with minimal fluctuation in the protein's RMSF, indicating the stability of the protein ligand structure. Although DHE is used as a migraine treatment that promotes cerebral vasoconstriction, we found that it had no significant effect on systolic blood pressure but provided significant neuroprotective effects, reducing the incidence, volume, and number of hemorrhages. Immunofluorescence analysis showed that DHE reduced leukocyte infiltration, ROS levels, and vascular smooth muscle cell apoptosis, thereby maintaining vascular integrity. Additionally, DHE treatment reduced the expression of NXP3 in neutrophils. These findings suggest that DHE may alleviate hypertension-related ICH and immune-inflammatory responses by targeting NXP3. In conclusion, our study suggests that NXP3 is a promising biomarker for ICH patients but requires further clinical validation. Additionally, DHE shows high affinity for NXP3 and can effectively alleviate the occurrence of brain hemorrhage. However, our study still has limitations. Although the mouse ICH model can induce vascular injury, it cannot fully replicate the chronic, multifactorial progression of human hypertension, especially regarding the long-term effects on the brain and other organs. Further human studies are needed to confirm the clinical relevance of these findings. Moreover, the clinical translation of NXP3 as a therapeutic target requires further investigation into its efficacy, safety, patient variability, and optimization of dosing and administration strategies.